We talked about the differences physiologically between males and females, and we talked about how the fiber type, the fiber sizes are different, but also the proportions, right? And how those proportions then influence metabolism is huge. So if you have more type 2 fibers, which is what we see generally again in males, generally depending on training status, then you're going to have more glycolytic pathways, which is reliance more on carbohydrates. When you have more oxidative fibers or type 1 fibers in females, you're going to have more capacity to utilize fats as a fuel.
Hi, Shani. It's my pleasure to have you on Evidence Strong Show. If you could briefly introduce yourself.
Hi, Alex. Thanks so much for having me. So my name is Shanie Landen. I'm a postdoctoral researcher at the Hudson Institute studying genetics and physiology. Other than research, I also compete over 800 meters. So it kind of gives me an extra interest in understanding how our bodies work when we adapt and when we exercise, and having a kind of molecular explanation for that.
That's perfect. So the paper you just published is titled Physiological and Molecular Sex Differences in Human Skeletal Muscle in Response to Exercise Training. Let's start with the most obvious and burning question, which is what are the differences in physiology between females and males?
If you kind of break it down into the different organs or the different tissues that we have, so males tend to have larger hearts, which means then larger stroke volume, higher cardiac output. That males have larger lungs and cross-sectional areas of their conducting airways. Males have more hematocrit and hemoglobin in their blood cells and more hemoglobin mass, stronger bones, more dense, and with what it means for muscles then and for adipose tissue. So males have a higher proportion, and I'll talk a bit more about this and kind of how this links in to the sex differences that we discussed in the paper, but males have a higher proportion, or tend to on average, of type 2, so the fast twitch glycolytic muscle fibers, while females, again on average, and it depends on training status, will have a higher proportion of the type 1 or the slow twitch muscle fibers, the oxidative ones. With adipose tissues, there's a different distribution of fat between males and females, with also males having relatively less fat percentage. So a healthy male on average versus a healthy female on average, males will have less fat. And that kind of sums up the components of the organs that we would think about when we're thinking about physiological performance. And what that then contributes to is males, quote unquote, outperforming females or having higher personal bests by 10 to 30%, depending on the event. And that will then contribute also to strength in many different aspects. So in females, relative strength between the upper body and the lower body is that females have more relative strength in their lower bodies compared to their upper bodies, while that phenomenon does not exist in males. So that just means, if you're squatting more as a female and not able to do as much bench press, like there's potentially a bit of a physiological explanation for that. And then the relative between males and females is that, again, on average, the male's upper body strength is 157% of the female's upper body strength. And for the lower body strength, it's about 60% more. So that just shows you that the discrepancy is much greater in the upper body between males and females than it is in the lower body. So yeah, despite those differences in strength and the capacity, males and females still, from what studies have shown, are able to increase their strength to the same degree, and so respond and adapt to the same degree, and that the differences in strength between males and females tend to appear more due to the muscle mass rather than the force generating capacity of each unit. So it's just the fact that there's more muscle fibers and they're bigger that can produce more force.
What are the molecular differences we know about?
So how I kind of think about this is that there's multiple levels of kind of the molecular profile. So we'll go through the central dogma the central dogma for genetics is that you have DNA, which is the same in every single cell of your body, essentially. And that DNA then gets transcribed into mRNA, the messenger RNA, and that will be different between different cells, between different tissues. And that mRNA then gets translated into proteins, and proteins go on to have their function within the cell and to communicate between other cells. There's then several layers of what we call gene regulation. So basically impacting which genes, so the DNA component, in the end get translated into proteins. And there's kind of many steps in which you can affect that process. So one of the ones that we looked at, which is a type of epigenetic modification, so epigenetic is kind of how our genes are interacting with their environment, with the environment in the cell. And that can come from a lot of different factors. It can be our diet, it can be our exercise, our sleep, the different toxins that we have that can interact with our DNA. And they don't change the sequence, but they tag on different things onto the DNA. So one thing that can get tagged on is what's called a methyl group. Then it's called DNA methylation, because a methyl group gets tagged onto the DNA that then determines how tight or how open the DNA is, and that can then decide which genes get then transcribed into the mRNA. Then we have another process that is the microRNAs, and that's different than the mRNAs, in that the microRNAs will find an mRNA and they'll say, oh, we're going to go attach to that one and degrade it. So that then influences how much mRNA is present in the cell. So that's another level. And then there's other, levels of gene regulation that I won't go into because I won't talk about them today, but there's other components as well. So it's not so, it's just those two. There are different levels in which the DNA then gets decided how much protein of that gene is left in the cell at the end, if that makes sense.
Okay, so in your study, you took biopsies of females and males, and you check what is the state of molecular translation in these cells, including the effect, so including the final product, the proteins that were produced.
Yeah. So the first thing we wanted to do was to just see at baseline, what are the differences between males and females in the muscle? Because at the time, it was not even ever looked at or known if DNA methylation even differs between males and females in muscle. It had been shown in blood, it had been shown in some other type of cells, but not in muscle. And then same thing with proteins. We didn't know on a kind of global level. So when I say global, it means that with our methodology, we used high throughput methods that are now very common practice, but it's technology that's come out in the last, couple decades or decade or two. And they basically allow you to look at all of the proteins or, thousands and thousands of genes at one time when it comes to DNA methylation as well. So it gives you kind of a genome wide is what we call it, or a picture of kind of everything that's going on in the muscle. And because those methodologies have developed over time. I say they've been around for a decade or two, but that's kind of like, it depends on which component we're looking at. And also the development with it has meant that it's become a lot more sensitive, a lot more accurate in the quantification. So what we were able to see is that just at baseline, before anyone does any exercise, there are thousands of genes that show different methylation in the muscle between males and females. And that then means that we expect them to have differences downstream, right? Differences in the mRNA. That was already known at the time. mRNA is kind of the most advanced in what technology has been around and for how long. And so it was already known that there are differences at baseline between males and females in skeletal muscle in their gene expression or their mRNA levels. And then with the proteins, we also found that just at baseline before exercise, there were several hundreds of proteins that were differentially expressed between males and females. So the functional unit protein at the end, if you just look at a male muscle and just look at a female muscle, you can tell that who it came from because the protein profile was very different. So that was to start it off going, okay, before we even exercise, how different are they? Yep, very different in both of those aspects. And then we looked at what happened after 4 weeks of high intensity training. So that was all on a bike, and it was all monitored to make sure that they're going at, a certain percentage of their VO2 max and a certain percentage of their lactate threshold. And it is quite like they're hard workouts. But of course, we also had people that came in with different training statuses. So you could have been kind of an untrained individual to a pretty fit individual. So that will also kind of, it was a limitation within our study because it will increase the variability of our results and potentially impact the findings. But in general, we were able to control for their VO2 max level. So that was kind of taken into consideration in our analysis. But in any case, what we found was that the proteins changed substantially after 4 weeks of training in the muscle, and they changed differently between males and females. So if you're a male, those proteins changed in that direction. And if you're a female, then different proteins changed in different directions. So that's what we found with the proteins. And then with DNA methylation, what we found, which was surprising to me at the time, or not what I was expecting, was that there wasn't a huge effect of exercise training on the DNA methylation status. It was slight, but not much, and there was no difference between males and females. It's like, yep, it changed a little bit, and what we can see looks the same for males and females. Okay, so then we got to thinking, if that's what happens with 4 weeks, which is, a small amount of time to be training, and we know, what adaptations lifelong training will bring are going to be a lot more robust, then we wondered, what about the cardiorespiratory fitness of these individuals? Because we had such a range and everyone performed VO2 max tests before and after they started, then we kind of had a fitness level for each one of them, which indicates lifelong training, cardiorespiratory fitness, right? VO2 max is a good measure of those. So we then used VO2 max to kind of give each individual a value, like this is how fit they are. The VO2 maxes ranged from about low 20s to, we had some very fit individuals that were like mid 60s. So we had quite a range. And we had a lot of individuals as well. There were 20 females and 45 males. So for, these muscle biopsy studies, which are kind of hard to get people to necessarily agree to give a piece of their muscle, it's relatively a decently sized study for this kind of endeavor. So yeah, what we found was that these fitter individuals, people with higher VO2 max, had a very different profile in DNA methylation than unfit individuals. So that indicated that lifelong training or your cardiorespiratory fitness influenced the DNA methylation profile in the muscle. And it wasn't different between males and females, which again was very interesting to me. It was like, if you're fit, this is what your profile looks like. Doesn't matter if you're a male or a female, even though there's baseline differences, and I can get to that in a second. And then what was also surprising to me is that we didn't find the same with proteins. So in proteins, we didn't see any effect of lifelong training on the proteins. So that was an indication to us that doing, an acute bout of training, right, 4 weeks, will potentially affect more the proteins, right? It has more of an acute effect. But that lifelong training, which is more, everything that's happened in your whole life, like your cardiorespiratory fitness, is more affecting your DNA methylation. That acute training was having a more substantial effect on the proteins, while the lifelong effect has more of an effect on DNA methylation, which is more of a stable process. So proteins can really fluctuate with depending on what you eat and what you do, but that potentially the DNA methylation is more transient and takes more time to change. And so we were kind of able to see that in both ways because of what we saw after 4 weeks and because of what we saw when we looked at the association between VO2 max. So since then, it was a really interesting finding to me to think that, your VO2 max or the training that you've done your whole life influences, something really deep in the cell, the DNA methylation of the cell, which it can change and is malleable, but it's less transient than the proteins. And so since then, we've completed another study which meta-analyzed other studies. So basically said, okay, we have our 65 individuals. We have some limitations in our studies. And since then, a lot of other studies had come out that had taken muscle biopsies and done VO2 max for those individuals. They didn't necessarily look for sex differences, but we were able to mine their data and meta-analyze it with ours. And we were able to confirm everything that we saw in our study, which is always comforting in science when what you found is replicable and you're not the only one who reached those conclusions. So being able to do that and replicate it in those other cohorts really strengthened what we saw, which is that the differences between males and females were evident at some levels of gene regulation and not at others, and that exercise training had a strong effect on the proteins early on, but later on with sustained training, more effect on the DNA methylation status in the muscle. So those were kind of our main findings. And microRNAs, we didn't go as much in depth as this, but we also found that the microRNAs changed differently between males and females after exercise. That was with an acute bout, not with the 4 weeks.
It sounds like methylation is important and it's early in the process of transcription. So potentially the impact is bigger, especially in the long run. This is something that the cell has to kind of plan in advance and put an effort to put methylation at the beginning of the process of transcription. And then things are happening. At the end, we have proteins. So with acute exercise, it seems like the end of the process can be manipulated pretty quickly. But the methylation part, maybe because it's earlier in the process, maybe it requires more from the body to be put in place. That's why it's affected slower. Am I close?
Yeah, I think that's kind of the idea that I have in my mind, is that it takes longer to have those effects because it's a more intricate process. With proteins, we know that you can break down, you have protein degradation, your protein synthesis. It's like a quicker, faster acting process. With DNA methylation, it requires, a bit more of kind of setup by the other proteins in the cells that need to interact to then tag that on. And then it's going to take time until, everything kind of flows downwards from that. I say time, everything in our cells happens pretty quickly, but yeah, essentially it is higher up the chain. It's like if you think about, you have a boss at the top, and if you want to get something happening and changing quickly at the bottom, if you get to send a message to the boss and wait for how long it takes to get to the bottom of the chain, that's going to take a while. But if you tell the person at the bottom straight away, then that's going to be much quicker in its effect.
Oh, I like it. Okay, so the methylation is important. I'm trying to gauge whether it's a good thing in relation to exercise and adaptation and performance. Is it a good thing or bad thing? Do we want more of it or less of it? Or is it just that it has to be in specific places? How should we think about it?
Yeah, definitely in specific places. So it's gotten really big now to look at the effects of aging, for example, on DNA methylation. It's a really hot topic right now, and it's something that my previous lab was working on a lot as well, and that I've been involved in. And what we kind of see is that aging has one effect on DNA methylation, and exercise has the opposite effect on those same sites. So it's like you have increases or decreases globally, yes, but it's more specific to the sites that it's at, and it's having its effect. So there's some genes that increase with methylation with age and with exercise, and others that decrease depending on the location in the gene. We know from other studies that there will be differences in how resistance exercise or endurance exercise impacts our molecular profiles in our muscle. I don't think that there's been enough studies out there for us to have a very clear conclusion on what kind of changes are different if you do strength training or endurance training, and how that impacts DNA methylation differently. Maybe there's something that's come out that I'm not aware of, but from what I know, there hasn't been a direct comparison to see the differences between those two training modalities. Whether frequency and duration, I mean, we know, with exercise training adaptation, it's all about intensity, frequency, and duration. So we can kind of presume, given that the changes that we were seeing were associated with VO2 max, then that kind of has intensity, duration, and frequency within it, right? So the more that you exercise and the higher your VO2 max is, that's where we saw that strong signature, that strong DNA methylation signature. So I would say, yeah, of course, it's the more intense and the higher that it is, the more that you're going to have this effect. And that's where we're seeing the impact on the DNA methylation. And whether there are other things that influence our DNA methylation, yeah, I mean, it's kind of a very blanket term to how our genes are interacting with the quote unquote environment or their environment. So there's a lot of different molecules or factors that will float around in our cells and impact which genes are methylated higher or lower. And things like smoking have a really strong effect on DNA methylation. Like we said, exercise. I'm trying to think what other studies have shown a strong cancer, disease. The things that are happening in our cells are very much influencing our DNA methylation. And you asked what's impacting our DNA methylation in a positive way. I mean, define positive. If you want it to look more like exercise, is that positive? If you want it to look less like aging, is that positive? I think it gets a little bit of a cloudy in that sense. But yeah, we do know that the things that we do, the toxins we're exposed to, also influence DNA methylation. But yeah, the main ones that have been studied and have been robustly shown are aging, smoking, cancer and disease, toxins, and now, yeah, exercise as well. I guess I could just add, it wasn't in our study because it was already known that, yeah, I think I did mention it, but that there are thousands of genes that are at the mRNA level, also different between male and female skeletal muscle. And there was a good meta-analysis that came out a few years ago that I can also link in the notes by another group that looked at all the studies out there that performed fossil biopsies and had males or females and trained. And they also saw that the mRNA were also changing differently after exercise, whether you're male and female. And there, I think they did group on different exercise modalities, if I remember correctly. So that could be to your previous question as well, with comparing the different exercise modalities, at least at the mRNA level. We didn't look at the other, I don't know of other studies that have looked at the other levels, but they did compare how jumped on another point of another important level of the central dogma, which is the mRNA, and that there have been a lot of studies on looking at the mRNA changes with exercise training. And there was a good meta-analysis that came out a few years ago that basically found all the studies that took muscle biopsies before and after training and looked and saw if there was a sex difference. And they did see that there were hundreds of genes that changed differently between males and females in muscle. So the mRNA level before and after training. And they did include different training modalities, I think. So they also had endurance and strength and made that direct comparison. So before what we talked about, we don't have a direct comparison from what I know of the DNA methylation changes and if they're different between strength and endurance, but we do know about mRNA levels changing differently depending on the training modality. And so yeah, I can link that in the link, send you that link as well if you want to put it in.
That would be awesome. So is it that we can see if we take genetic material from skeletal muscle of a person, random person, we can guess whether they are athletes or not and potentially what type of sport they are doing?
I thought you were going to ask about the male and female aspect because that part's super interesting. There's actually a really cool paper that found
That is my next question.
next one. So I'll answer that one first just because it's more definitive of an answer and I think it's fascinating. But a paper that came out a few years ago tried to predict if we look at the mRNA in a tissue, can we predict if it's male or female? And they knew if it's male or female, so they could check if the prediction was right. And they looked at all the tissues, 40-something tissues in the body. And it's using what's called the GTEx portal. So it's a large publicly available data set that has hundreds and hundreds of people that have donated their tissues after they've passed away. And so it's quite robust, the findings. And they found that besides breast tissue, skeletal muscle predicted sex the best out of any other tissue. So that just shows you, just how that's, we call skeletal muscle one of the most sex biased tissues in the sense that it really is differentiated between males and females compared to, brain tissue, liver, kidney, any other tissue that was in there. So yeah, which makes sense given how hormone responsive it is and that the hormones will really differ between males and females. And I had a whole bit I was going to explain to you about hormones versus genetic. Yeah, so that's to answer whether muscle can differentiate. We can differentiate based off of muscle if it's males and females, and whether we can differentiate their training status. That's a really good question. Maybe that's a future paper that we work on. Absolutely, because there's lots of kind of machine learning algorithms out there to try to predict those things. And if we had the VO2 max of those individuals, if we were able to predict how fit they were, that's an awesome question. I'm going to steal that. Let's see if there's a student that wants to work on that.
That's awesome. This we have to know if we can predict pretty robustly whether the tissue, the muscle tissue, is from a female or from a male. Why they are so different? What is happening from the genetic point of view and then through physiology that we end up with so different tissues?
So that's something that I tried very hard to answer during my PhD and in my thesis and was limited in my ability to answer that because typically females have XX sex chromosomes, so 2 X chromosomes, while males will have XY chromosomes. And that then sets up many differences in our cells, in our DNA. And what I mean by that is, so that's kind of the first difference that we see, at day 0, at conception, the embryo is either XX or it's XY. And if everything goes kind of typically and there's no variations to the sex development process, which is the lab that I work in now, is very focused on sex development and on the different variations that happen to this process. And I can speak to how that kind of relates to my previous work as well. But those first differences are going to set you up for all the differences that come downstream. So if you have XX or if you have XY, it basically takes your gonad development. So we start with a bipotential gonad, which means it has 2 potentials. It can go on to be ovaries and it can go on to be testes. And you need to have a few key players present or absent in order to kind of go down those 2 pathways, one of those 2 pathways. And so having XX, if everything goes normally, will then develop into ovaries, which then means you have high estrogen, high progesterone, and lower testosterone levels. Or if you have XY, that typically will take you down the other pathway in which you develop testes, and you have higher testosterone relatively and lower estrogen and progesterone. And so then there's kind of then thought of as two main drivers of the sex differences that we see. So everything that we talked about until now, the performance, the physiology, the proteins, the gene regulation. It's thought that those two are driven by those things are driven by those two components. The difference, the fact that you have XX and XY, and the fact that you now have different hormone levels for the rest of your life. And those two things will impact what goes on in the cell. And so it's kind of impossible to break apart those two drivers because they come together, right? If you have XX, you have that hormone profile. If you have XY, you typically have that hormone profile. So how will you say if the difference that you see is due to having different sex chromosomes or different hormone levels? Because, well, they come together, you can't break them apart. So that's what a big project that I'm currently working on in the lab that I'm in now, which is that the lab that I work in before I came in developed in collaboration with a couple labs in the US, what's called a 4-core genotype model. So it's a model that's existed in mice, again, by our collaborator at UCLA for over 20 years. And when I came in, they had just finished producing a rat version of this model. And what this model is, and why it's called the 4-core genotypes, is that we end up having 4 genotypes. One that's, a typical XY male, a typical XX female, but then we have XX males and XY females. And what that allows us to do is to break apart the impact of the sex chromosomes and the hormones, because now they're not linked together.
Shani, just to make it clear. So when you say XX female, that's XX genetics and female hormonal profile. If you say XX male, you mean XX genetic profile, but hormonal profile is the profile of a male. Is that
exactly right? Yeah, yes. So when I say male or female here, it just is describing phenotypically or the gonads. The moment the gonads are testes, have high testosterone. The moment the gonads are ovaries, you have high estrogen. So yes. So yeah, that model has been used in mice for decades now to understand the drivers of a lot of different sex differences. So one, for example, is that what was found is that the X chromosomes are what strongly influence adiposity. So animals that had 2 X chromosomes, regardless of their hormone levels, were fatter and ate more. And so it was kind of a, revelational in the sense that, we see so many of the differences. It was commonly thought that so many of the differences that we see between males and females are due to hormones. And we know that hormones have such an influence on fat cells and on adipose tissue. But yeah, that study had kind of shown that there's also a role for the genes that are on the X and Y chromosomes that's been previously underappreciated. So there are a lot of studies that have come out showing there's a specific gene on X or a specific gene on Y that can influence a specific trait or a specific disease. So yeah, it's understanding that there's also now this level of, well, if you have 2 X chromosomes, it means that you have a different level of genes expressed in those chromosomes if you're a female. And if you have an X and Y, then you have different genes there as well in a male. So there's those inherent differences. So you asked, to bring it back to the exercise adaptations and what we were seeing. I tried my best to answer that question with the human data that we had, which is, well, those two things are linked. So I can't really pull those components apart. We did as much as we could, and we did see that if you looked just within females, and we did have all their hormone levels measured in their blood, their testosterone, their estrogen levels, their SHBG, we all measured, and we did see that within females, because again, we couldn't compare males to females without having those two things intertwined, right? The genetics and the hormones. But within females, we saw that hormones were impacting the DNA methylation level. So the level of hormones. And I mean, there's not a great variability in testosterone within the female. So that's not a huge finding. But with estrogen, it's really variable because it's changing throughout the cycle and it will be different between people. And while we did collect blood and muscle from people always in their early follicular stage, so the first 7 days of their cycle, to try to minimize that variability, there will inherently be variability because that wasn't also the goal of the study. So it wasn't, super tightly controlled as it could be. It was just as tightly controlled as we kind of needed it for the bigger question. But we do see that hormones influence the DNA methylation levels preliminarily from what we found, but that further work is needed, which is kind of what my lab is working on now in collaboration with another lab, is to understand what is driving the sex differences in the muscle. And I can tell you what we found so far. I'm still preparing that manuscript and don't have super firm conclusions yet, but what I'm finding so far that's really interesting is that there is a really strong effect of the gonadal hormones, so the testosterone, estrogen, progesterone, on the mRNA levels in the muscle of these rats. But there is still some component that's influenced by the sex chromosomes. So we've kind of looked at a bunch of different tissues and tried to see, okay, what's having an effect of the in the mRNA levels in these different tissues? Is it the sex chromosomes or is it the hormones? And for most of the tissues, it's been the hormones, but not for all. And muscle is one of the ones that hormones seem to be having a much stronger effect. But again, that there is still some effect of the sex chromosomes, which is super interesting. And we're still going through kind of each gene, each mRNA and seeing, okay, what's this one? What's its function? Why is this one driven by hormones? Why is that one driven by the sex chromosomes? So we're still kind of piecing that together. And the best that I could kind of do in my PhD with the human data, because again, that was rat data, and the best I could do in my PhD with human data was to kind of see if there's hormone responsive sites around the DNA methylation differences we were seeing. And we did see that androgen receptor was nearby those sites, which indicates that likely there's a kind of a hormonal explanation for some of the DNA methylation differences that we were seeing in the human muscle.
If we know that there are differences between females and males, and some of the differences are due to genetics, some of them are due to hormones. If there is any relation between how hormones influence genetics, I would be interested to know.
Okay. Yeah. I mean, I think it's a fascinating topic, so I kind of had it in here. So how progesterones, estrogens, and testosterone work is there's kind of two signaling pathways, but I'll focus on the main one. And the one that's kind of the most canonical one is that hormones have receptors. We have testosterone that's floating around in the cell, and it needs to bind its receptor. That's how it exerts its action. And when it finds its receptor and they're together, they then enter the nucleus where the DNA is held, and that receptor can either directly or indirectly bind the DNA at specific sites. So there's specific, what are called hormone responsive elements. So different sequences that will say, okay, we recognize when there's an androgen receptor and it will bind here, and it will then directly impact which genes get transcribed. So which mRNA then come out from that interaction. So that's how hormones are really directly, or we say indirectly because there's sometimes other proteins that are involved, but we can say directly or indirectly changing which genes are getting transcribed into mRNA. So that's kind of how hormones exert their effect. And yeah, so basically they're kind of seen as what we call transcription factors, because they are a factor, right, in transcription. They can bind the DNA. The receptors can bind the DNA.
What the future is bringing.
Yeah, I think with every scientific question, the moment you answer it, you just kind of come out with more questions. So there's always more questions to answer. And a few that I kind of had left after our work, or with our work so far, a big one is the influence of the fiber types. So I didn't really, I didn't go into this. I probably should have, and is more interesting to your audience than all the molecular details that I gave. But I will add in that maybe you want to put it in earlier on, that it's not really a summary. It's something I meant to mention. I just realized is that what we know about, and we talked about the differences physiologically between males and females, and we talked about how the fiber type, the fiber sizes are different, but also the proportions, right? And how those proportions then influence metabolism is huge. So if you have more type 2 fibers, which is what we see generally, again, in males, generally depending on training status, then you're going to have more glycolytic pathways, which is reliance more on carbohydrates. When you have more oxidative fibers or type 1 fibers in females, you're going to have more capacity to utilize fats as a fuel. So it has been shown multiple times that in endurance training, females will have a bit more of a reliance on fats as a fuel, while males will have a bit more of reliance on carbs as a fuel. Of course, they're both relying on both throughout training, but the shift is a little bit different given that we have different fiber type proportions, right? Type 1 versus type 2 within a male muscle or within a female muscle. So that then is a huge question left to answer with what we've seen. So are all the differences that we're seeing, in DNA methylation, in proteins, in mRNA, is that partially or very largely driven by the fact that we actually have different distributions of type 1 and type 2 fibers? And again, I tried to answer that as best as I could because we did check for the fiber type proportions in all of the individuals, the 65 individuals that we had in our study. It's the data I was analyzing the night before my wedding. But anyways, no, we won't get into that. Yeah, so that's a huge question left to answer is, how much fiber types are influencing the molecular profiles that we see. And it's something that a colleague of mine did his PhD on after I completed my PhD. So he has some papers coming out on that, and that work is still ongoing. Preliminarily, or what he's finding so far, is that there is a component of the proteins that is largely impacted by the fiber type differences between males and females, which is also what I was finding in DNA methylation, that there's a really strong signature of DNA methylation in type 1 fibers or in type 2 fibers, with, again, the limited ability of my study to answer that question because it wasn't designed for that. And there have been studies out there showing just how different the kind of molecular profiles are, right? The proteins and the DNA methylation, and how they are different between type 1 and type 2 fibers. So that's a big part to take into consideration with things left to do. Another part, which is what we touched upon, which is what's driving those differences, which can be answered in a multiple different ways. One is what we're trying to do with the rat model. The other is using, indeed, gender affirming hormone therapy individuals and seeing what hormones will do to their muscle. And yeah, there's kind of a few different ways we could try to answer that question. And the other thing is really just understanding the different levels of gene regulation, because that'll paint a bigger, kind of more comprehensive picture of what is happening with exercise adaptations. And, if we were seeing differences between males and females on the protein level, but not at DNA methylation, and we're seeing it at mRNA, what's happening with everything in between? Like, what is the mechanism behind it? And while we try to answer it, in a few different ways, it's, as we kind of hopefully alluded to, is pretty complex. And there's a lot of different players. There's a lot of different ways to regulate which proteins end up at the end of the day having their function. So kind of at which level is everything happening? Where is exercise exerting its effect? Where are the sex differences coming in? Are they earlier on? Are they later? Are they across the whole process? Yeah, just the complexity of the gene regulation, I think, is still a lot of questions left to answer there.
That's awesome. Thank you so much for sharing what we know so far. Let's finish with some advice for coaches. So if you could bring it all together and compress it, would you have anything to say that coaches should know about the differences between female and male muscles?
I think a better understanding of what is going on in our bodies, in our muscles, contributes to a better understanding of kind of how things work. I don't know, how you want to think about life and training. And I mean, other than the kind of basic ones that we know, which is, females have, greater relative lower body strength and upper body strength. And I don't know if that's something to take into consideration when you're planning, your load and the rep, the load that you're aiming to hit for your different reps. I think a lot of it also comes down to practice and to individual experience. And I think while, every individual is going to be different, we're looking at males and females as a whole, but of course there's so much individual variability as well. So it's hard to kind of have these concrete, conclusions on this is what you now should do. But I think it's just the more we can understand what's happening molecularly, the more we can just get a bigger, more clear picture of, what exercise adaptations are looking like. And, what is that lactate burn at the end of a 400 meters? What does it mean on a molecular level? Yeah, regarding looking at males versus females and how to really take that into consideration. There's a few examples come to mind. Like, I think males tend to have a larger difference between their first and second lap in an 800 meters. So, right, you run and then you get really tired and continue running and you slow down a bit. Males tend to slow down more compared to females, which in my molecular mind makes sense, because if we know that females tend to rely more on fats and kind of have relatively enhanced endurance compared to males, in the sense that, they're able to kind of rely on that process more, then it kind of makes sense to me that they're able to run a more even pace, right? If you're relying on fats and your oxidation, then you're relying more on your endurance capacity. So you're going to be a bit more even when you're thinking about males in the 800. And if you're slowing down, then you've relied a bit more on speed to get you there, and then you've slowed down, so it's kind of hard to put into. I feel like I didn't explain that really well, but
What I'm hearing is that females run slower in general than males, but they are able to keep the tempo better because they are slower, probably because of the same reasons that they are able to hold the pace. So the genetics are different, the muscle mass is different, the fueling is different.
exactly.
And they are also some mental, females and males mentally are a little bit different too. And there are some factors in the sports that will benefit potentially from that too. So lots of things, very complex, but I get you where you're going with it.
Yeah. Yeah. I think depending on the exercise that you're interested in or that you're doing or that you're involved in, you can potentially think about the different components of this in different ways. So my head goes to that and realizing that, those are some of the differences we see in the 100 between males and females is the pacing. And that can kind of be explained by the understanding of the metabolism and the molecular components of it. So yeah, depends on what you do. And then, same with strength. If we're looking at upper versus lower body strength, then it's another thing to understand. Yeah, in that sense is the best way I can kind of think of how to relate this to practice.
Beautiful. Two short questions to finish. The first one is, what is your favorite exercise? You already said 800.
Yeah, I was gonna say I don't think I should stray away from that. But yeah, if you were to ask me like my specific, the 800 is a delicate combination between endurance and strength. So kind of get to do everything from speed training to speed endurance to strength, power strength training. So a bit of everything, which I love that aspect of it. Probably my favorite workouts is like 200-meter hill repeats. There's just something to making it to the top of the hill. You and your glutes are on fire and just you feel on top. So yeah, it's probably my favorite workout. But yeah.
Awesome. And the last question is where people can find you online if they want to follow your work and learn more.
I'm not the most active. I do have LinkedIn, so if you just search my first name and last name, I'm the only one. But yeah, so I'm trying to post more and get more exposure to our work. So through LinkedIn would probably be the best way.
Thank you, Shani. It was a pleasure.